Our final speaker is Orlando Yarborough, who's now at Yale University School of Medicine. I want to thank, by the way, all of our speakers for keeping to our time schedules. I think we're pretty well on pace to finish on time at 11 o'clock, or I'm sorry, 10:30. And the title of Orlando's talk is Modulators of Blood Pressure, Discovering Novel Proteins in the Wink-4 Kinase Signaling Cascade. Let's get some talk up and running. Good morning.
Good morning.
High blood pressure or hypertension is a particularly complex disorder reflecting the complexity of the blood pressure trait. Hypertension affects 1 billion people worldwide, approximately 1 in 4 adults in the U.S. It is a major risk factor for mortality through heart attack, stroke, end-stage renal disease, and congestive heart failure. Though despite being such a major public health challenge, its pathogenesis is largely unknown and its treatment suboptimal. The complexity of blood pressure is reflected in this pioneer's wiring diagram, suggesting that the regulatory centers for blood pressure to include heart, kidney, brain, vascular system. So if a patient comes into your office with high blood pressure and you're using this chart, do you target your treatment to the brain, to the kidney, to the vascular system? Human genetic studies in our lab have identified the kidney to be the main regulatory center. The kidney maintains water and electrolyte balance through filtering blood into the nephron, the functional unit of kidney. Our lab today has identified mutations in 10 genes which elevate blood pressure, and an additional set of mutations in 10 genes which lower blood pressure. And strikingly, all of these genes function in kidney nephron, regulating salt reabsorption. One hypertensive disorder, pseudohypoaldosteronism type 2, or PHA2, is an autosomal dominant disorder, meaning that each affected parent has a 50% chance of transmitting that disorder to each child. Other characteristics of PHA2 are high blood pressure, elevated blood potassium levels, and all of the phenotypes are chloride dependent. Our lab identified mutations in serine threonine kinases Wink1 and Wink4 to cause PHA2. The mutations in Wink1 are deletions in the first intron, and the mutations in Wink4 are single base changes in DNA. Our lab went on to investigate the molecular components that link Wink4 to the regulation of blood pressure. In the kidney, Wink4 expression is localized to distal nephron segments. Our lab has shown that wild-type Wink4 inhibits the sodium chloride cotransporter NCCT, which reabsorbs sodium chloride from the urine back into the blood. Wild-type Wink4 inhibits the sodium channel ENaC, and it also inhibits the potassium channel ROMK, which moves potassium from the blood into the urine, excreting it from the body. PHA2 disease mutant Wink4 loses its inhibition of NCCT, and it loses its inhibition of ENaC, which leads to increased NCCT activity, increased ENaC activity, causing an increase in sodium chloride reabsorption given the hypertensive phenotype seen in the PHA2 patients. And interestingly, the same Wink4 PHA2 mutant increases in its inhibition of ROMK, leading to decreased ROMK activity, decreased potassium excretion given the elevated blood potassium levels. Furthermore, both wild-type Wink4 and the PHA2 Wink4 causes the reabsorption specifically for chloride. The process for chloride reabsorption is poorly understood, and though we know some of the molecular targets of Wink4, 2 questions remain unanswered. Largely, what are the upstream regulators of Wink4, and what are the molecular steps which link Wink4 to its final targets? I'm taking a gene expression approach, overexpressing Wink4, and looking specifically for RNAs and specific proteins which change. And given changes, I cross-reference the RNA and protein data sets in a joint genomic approach that increases the power for identifying novel components involved in Wink4 signaling. For my genomic approach, I need a model system that is simplified that allows me to study Wnt4 function in kidney. And I'm using a dog kidney cell line, MDCK cells. The next diagram is going to outline the nephron epithelial tubule, and it reflects many of the properties of the MDCK cells. In the nephron, the epithelium is one cell layer thick, polarized with defined apical and basolateral surfaces, and the reabsorption of chloride that is due to Wnt4 and PHA2 Wnt4 occurs through a paracellular route, which is flux between cells, known as paracellular ion flux. And the MDCK cells I'm using are the best characterized polarized epithelia for studying paracellular flux. I engineered these MDCK cells to overexpress Wnt4 in a user-controlled fashion. Now, to make this model system useful, I need a biological and physiological measurement for Wnt4. So to find one, I grew MDCK cells with and without Wnt4, plated them onto a membrane where I can measure the electrical resistance across the monolayer, and that reflects the paracellular permeability for that monolayer. Looking at the percent decrease in electrical resistance plotted on the y-axis, focusing on the center column, when I overexpress wild-type Wnt4, there's a significant decrease in electrical resistance as compared to monolayers without overexpressed wild-type Wnt4. Similarly, when I overexpress PHA2 Wnt4, there is also significant decrease in electrical resistance as compared to the uninduced monolayer. And furthermore, the percent decrease due to expression of PHA2 Wnt4 is greater than the decrease due to expression of wild-type Wnt4. So now I have a biological effect for Wnt4 and a different biological effect for PHA2 Wnt4, and I'm looking to identify genomic coordinates which reflect these effects. Using comparative proteomics, I survey all the proteins in a cell, but I only give attention to those proteins which are changed between two experimental conditions, namely with and without Wnt4. In total proteomics, I'm looking to discover novel proteins involved in Wnt4 signaling. In targeted phosphoproteomics, I'm focusing my attention only on the known targets of Wnt4, looking to characterize their phosphorylation state. In total proteomics, I'm using differential fluorescence gel electrophoresis, or DIGE, where I grow cells with and without Wnt4, I harvest all the proteins, and then I independently label each sample with a different color fluorescent dye. I then combine the samples and run them simultaneously on a 2D gel, coupled with mass spec for simultaneous analysis. I'm able to identify proteins and statistically quantitate changes in protein abundance, localization, and phosphorylation. And looking at targeted phosphoproteomics, I go back to the nephron, where the paracellular flux of chloride is governed by a physical barrier known as tight junctions, characterized by a family of proteins, claudins. Claudins are differentially expressed along the length of the nephron, and boxed in blue are those claudins that I have detected in my MDCK cells. In targeted phosphoproteomics, I use a SciLight technique where I can grow cells in culture, labeling each protein in a cell in each cell. The wild-type Wnt4 cells are cultured under normal conditions using natural or light amino acids. To the Wnt4 PHA2 cultures, I add heavy amino acids, which will be incorporated into every new protein synthesized. I then harvest all the proteins, and I mix the samples in a 1-to-1 ratio. I purify the claudins, I digest the proteins into peptides, and I run them over a titanium dioxide column, which enriches specifically for phosphorylated peptides. And then, coupled with mass spec, I can get the ratio of claudin phosphorylation between the cell population with Wnt4 or with PHA2 Wnt4. Using my joint genomic approach, I'm able to increase the power for identifying effectors or identifying targets in the Wnt4 signaling cascade, and this will also help to further elucidate the molecular pathophysiology of essential hypertension as seen in the general population. Thank you. the changes that happen in the kidney because we've done that research. So right now I'm using the MBCK cells, but I will go back into mice and look there. Yes?
So you said you're looking for novel targets. Is there any information on what Wink already phosphorylates?
Yes, so we know that Wink does phosphorylate some claudins. Wink has been shown to phosphorylate claudins 1 through 4. There are about 20 different claudins. And for one of the claudins, a specific phosphorylation site is known, but for the rest of the claudins, the phosphorylation sites are not known. One thing about Wink4 is that it's expressed in the kidney, but it's also expressed in diverse chloride-expressing epithelia all over the body, and it is not known those targets there. Yes?
What do you mean by phosphorylate?
Oh,
Like,
yeah.
Like you're taking phosphor and you're putting it on something, or what?
Good question. So phosphorylation is a modification that can be added onto proteins in order to influence the function of that protein. So sometimes phosphorylation is added to turn a protein on or off, or to cause degradation of a protein, or to take a protein that's in the cytoplasm and insert it into the cell membrane. For my phosphoproteomics, I'm able to take a protein and using mass spec, not only find out what the amino acids are, but to ask which amino acids are phosphorylated. And when I get a protein that has a sequence of amino acids, I can say that all these amino acids within this protein peptide are phosphorylated, and I can correlate that with my physiological parameters saying, when we see an increase in electrical resistance or when we see an increase in chloride reabsorption, these proteins are phosphorylated in this state. When we don't see an increase in chloride reabsorption, these proteins are phosphorylated in this different. Does that answer your question?
So in that case, by phosphorylate, you mean turn on, like these proteins. When you do this, these proteins turn on, and when you do something else, they don't turn on. That kind of thing.
Yes, phosphorylation can be used for that, but specifically phosphorylation is just kind of a structural modification that is made to proteins where a kinase adds a phospho group, a phosphopeptide or a phospho group onto the amino acids of a protein. Yes?
How is phosphorylation in this case different from something like ADP, ATP? I understand that doesn't last very long, so probably won't be able to measure that. How is this different?
Okay, so the kinase, Wink4 is a kinase, and it's able to take a phosphorylation group and put it onto its target protein, and it's not known, we would call it phosphorylation dynamics, but it's not known how long that phosphorylation lasts on that protein. So whether it's a quick event or whether it's a slow event. We need it to be slow enough to be able to capture it in our analysis. So literally, the experiment is done within a couple of days maybe 2 days, where I harvest the protein from the cells, saying that they're in their native phosphorylation state. I do minimal manipulations and go right to mass spec to try to capture their phosphorylation state, and I inhibit any other proteins within the cell that may cause dephosphorylation or extra phosphorylation of that protein. So I try to capture it in its native state, and hopefully that will minimize any changes that might happen.
So even if the phosphorylation of that is very transient, at any given time there should be some subpopulation that has of the protein that is phosphorylated, so as you said, you can capture that snapshot.
Yes, yes. And then we can always go and isolate the proteins and stabilize them so they're never dephosphorylated, and take a phosphorylation-specific antibody that can recognize specific amino acids that are phosphorylated. So that's another way of doing it without using mass spec. Thank you.
Okay, so